Hello, everyone, and welcome to today's webinar, Accelerating Lentiviral Process Optimization with Multiparametric Macro Mass Photometry. My name is Delia Osborne with LabRoots, and I will be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Refine. To learn more about our sponsor, please visit www.refine.com. We encourage you to participate today during today's session by submitting any questions that you may have. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of our presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. I'd now like to welcome our speakers for today's webinar. Hena Divanovic, Application Scientist at Refine, and Dr. Qian Liu, Head of Viral Vector Process and Innovation at Minaris Advanced Therapies. Dr. Liu, you may now begin your presentation.
Thank you for the kind introduction. I think I will just start the presentation today by going to our agenda. First of all, I will give you an introduction of Minaris Advanced Therapies, and then followed by our lentiviral vector platforms and some process development work that we have been doing. I will highlight some challenges we have encountered during the process development, and then introduce you to my co-presenter to introduce the advanced lentiviral vector, how they are advancing the lentiviral vector in process analytics. And then Hena will continue with the how micro mass photometry work, and give you some examples of how they help tackle the challenges. So first of all, I would like to just give you some introduction about Minaris. Minaris was formed in 2025 by bringing together two long-established leaders in cell and gene therapy development, manufacturing, and biosafety testing. One of these organizations is Advanced Therapies. So that was the US and UK operations of the WuXi Advanced Therapies. And this organization brought deep expertise in contract development, manufacturing, and testing. In addition, we also have Minaris Regenerative Medicine, originally progenitor cell therapy and EPSETH by pharma. So they contributed to more than 25 years of specialized cell and gene therapy CDMO experience. So by uniting these two complementary leaders, Minaris is now a fully US-owned leading cell and gene therapy CDMO and multimodality biosafety testing provider. We have divided our team into two business units accordingly, namely Minaris Advanced Therapies and Minaris Advanced Testing. Minaris Advanced Therapies, which is the business unit I work in, is the CDMO business unit of Minaris. We solely focus on supporting the CGT developers from concept to commercialization, offering a truly end-to-end service. And we are able to offer this end-to-end service thanks to our deep hands-on experience across CAR-T, NK cell, iPSC, MSC therapies, and all major viral vector types, including AAV, lentiviral vectors, and adenoviral vectors. Specifically on the viral vector side, we can support cell and gene therapy developers from the basic but very critical plasmid stage. We offer the off-the-shelf lenti and AAV packaging plasmid systems and customized plasmid design service to ensure high vector yield, desired transgene expression level, and good regulatory compliance. We also offer adherent and suspension platforms for industry-standard viral vector manufacturing processes and have developed proprietary platforms like TESSA for AAV and SOFLEX for LVV, which are available for licensing and can significantly improve manufacturability and reduce cost of goods. I'll talk more about the Solflex LVV platform in later slides, as we'll mainly focus on lentiviral vectors today. And you may ask why we're focusing on lentiviral vectors today. So if we have a quick look at the approved CGT products and the ongoing clinical trials in this field, we can see that lentiviral vectors are being used in 2 out of the 4 blockbuster CGT products, approved products, and it's also being used in about 40% of CGT clinical trials. It is such a widely used gene therapy vehicle in cell and gene therapy field, thanks to some quite unique advantages of this viral vector type. First of all, lentiviral vectors are able to transduce both dividing and non-dividing cells and can mediate a long-term durable transgene expression. It has a relatively large packaging capacity, about 9 kb, for like a wide range of transgene cassettes. And it is safer than the gamma retroviral vectors, as it has reduced the preference for integrating in promoter or enhancer regions. And also the self-inactivating LTRs are being used in the 3rd generation lentiviral vectors to further reduce the risk of generating replication-competent viral particles. So at Minaris, of course, we want to support the cell and gene therapy developers in the lentiviral vector production side. So our team have spent a lot of time and effort developing our lentiviral vector manufacturing platforms to ensure we produce the vectors at high yield and good quality. So this slide is just to give you a quick overview first. We have developed both adherent and suspension production processes to fulfill different preferences of CGT developers. We also have proprietary lentiviral vector packaging plasmids and also the Solflex stable cell lines, which enable higher productivity and improved process consistency, enabling reduced cost of goods and a better manufacturability. Our process development team pre-developed the upstream and downstream platform processes to ensure optimized performance and consistency. And we provide comprehensive support to advance cell and gene therapy programs at all stages, from discovery and development stage to clinical development and to finally commercialization. Now, let's just have a little deeper dive into the standard suspension cell line platform at Minaris. We developed the clonal suspension HEC293 cell line from a polyclonal parental cell line. After isolating, expanding, and testing over 3,000 clones, we identified the best one as our viral vector platform cell line. It's a HEC293 cell line without the SV40 large T antigen, so it is safe and suitable for manufacturing viral vectors for both ex vivo and in vivo applications. We have created GMP-grade master cell bank and working cell bank for this cell line, and the drug master file is also available to support regulatory submission. The viral vector production processes using this cell line have been established and can be scaled up to 1,000 liters. In addition to cell lines, of course, packaging plasmid system is also a critical part in the equation. Our current lenti packaging plasmid set was developed by Legacy Oxygen team. It's a 3rd generation self-inactivating system with codon optimization and minimization done to improve lentiviral production efficiency. We also optimized the sequences to reduce the intercassette homology to further improve the safety profile. We actually compared our lentiviral packaging plasmid set with a few competitor products on the market. And from the head-to-head comparison, we have observed that the lentiviral vectors produced from our plasmid set had the highest production titer and the best particle to infectious titer ratio, so the best P to I ratio. So built up on the high-performing cell line and plasmid set, Minaris' standard 4-plasmid lenti production process is offering an industry-leading solution to cell and gene therapy developers for lentiviral vector-based therapies. However, we do also acknowledge the limitations in the nature of this production approach. First of all, it requires 4 plasmids, and when we're talking about GMP grade manufacturing, it needs like 4 GMP grade plasmid batches, which could cost a fortune. And the multi-plasmid transfection process is complex and has relatively high process variation, which reduces process robustness and limits the scalability. So our scientists would like to improve the production approach by developing stable cell lines to manufacture lentiviral vectors, hence reducing or removing the plasmid costs and enhancing the process robustness and scalability. We firstly integrated the envelope and packaging elements, namely VSVG, GACPO, and REF, into our clonal Hex93 cell line to generate Soflex packaging cell line. Of course, the VSVG and GACPO are under inducible control due to their cytotoxicity, and they are only activated when the inducer is added to the cell culture system. The packaging cell line enables single plasmid transfection for lenti production, so this reduces plasmid costs and transfection complexity. Then, based on the packaging cell line, we integrated the lenti transfer plasmid into the cell line to generate Soflex producer cell line. And this cell line offers the transfection-free lentiviral vector production process with plasmid and transfection reagent costs eliminated completely. It also boosts the viral vector production process robustness and scalability to the next level. Let's just have a very quick look at the stable cell lines lenti production performance. So on the graph on the left-hand side, you can see that we have tested lentiviral vector production for a quite wide range of transgenes in the 3 lentiviral production platforms we offer. So these are the 4-plasmid transfection process, packaging cell line, and producer cell line. And you can see that in most cases, the packaging cell line showed comparable or slightly higher titers than the 4-plasmid process. And the producer cell line, in most cases, showed higher production titers than the other 2 processes, so the highest productivity. And not only that, the producer cell line also showed the lowest batch-to-batch variation among the 3 platforms, as you can see in the graph on the right-hand side. And this is demonstrating it has the very good promising robustness, promising the process robustness. The performance data shown in the last slide just confirmed the advantages of using stable cell lines, especially the producer cell line for lentiviral vector production. The producer cell line, it reduces cost of goods and de-risks manufacturing process and improves scalability. So it offers a ideal solution for therapies that require a large quantity of lentiviral vectors, such as the uptrending in vivo CAR T therapies and the other types of lenti-based in vivo therapies, as well as the ex vivo therapies for more prevalent diseases like autoimmune diseases. Our next slides will be focusing on the producer cell lines from the process development perspective. The producer cell line, it also actually allows some extra process innovation that might not be feasible for a transfection-based production process. And this could potentially result in further improvements in vector yield, cost effectiveness, and manufacturability. Here on this slide, I'm showing a representative lentiviral vector production process, starting from the upstream production in a stirred tank bioreactor, followed by a clarification step, AEX purification, and TFF concentration, and the sterile filtration as the last step. So all of these processes to purify and concentrate the lentiviral vectors. In each step, there are some key parameters to test and optimize in our a general process development process. Some of these parameters are applicable to all the lentiviral vector platforms, such as these downstream parameters in general. But there are also some optimizations that are quite unique to producer cell line. For example, if we want to test extremely high cell density, if you want to test additives or enhancers, that might not be compatible with transfection, or if you want to try to extend the harvesting window to multiple days or even weeks, all of these optimizations can only be tested in a transfection-free process, and this is really showing some process advantages of the producer cell lines. And before we start any unique process innovation, we firstly wanted to test the producer cell line in our standard platform process to check the performance. And as you can see in the graph, without any specific optimization or changes to the process, the LV production from the producer cell line at 3-liter scale resulted in about 1E8 transducing units per mil upstream titer. This is very comparable to the small-scale production data. And the downstream process step recoveries were all as expected as well, with the P/I ratios kept consistently below 100 throughout the process. This result showed that the producer cell line adapted very well to the platform process, demonstrated the cell line's good scalability, and set up a good baseline for additional process innovation and optimization. Our team explored quite a few key parameters in producer cell line-based lenti production process, including those unique processes to produce the cells and some general parameters. And now I'm just going to give you a couple of the examples. In this example, we were trying to reduce the autotransduction level during lentiviral vector production as a way to improve lentiviral vector upstream production yield. As you probably know, that during lentiviral vector production process, the produced lentiviral particles could retransduce the production cells, which we call autotransduction, and this obviously would reduce the vector yield coming out of the production process. There are different ways to reduce autotransduction from various perspectives. From engineering side, you could modify the vector envelope or the production cell line surface to make the production cells not transducible by the vector envelope. From process side, I've read groups changing conditions like pH to reduce autotransduction. And in our case, we wanted to test a range of additives and reagents and see if any of them managed to improve lentiviral production yield through reducing the autotransduction level. And using the producer cell line allowed us to test a wider range of additives, as some of them may inhibit transfection-based process. Out of the tested reagents, we identified an additive, we call it additive X, which increased the lentiviral production titer from producer cell lines by about 2-fold. And indeed, we also saw a significantly lower level of autotransduction from the producer cells treated with additive X. We then managed to apply the same additive to packaging cell line and the 4 plasmid lenti production processes. And as you can see from the graph, we have observed a similar effect in packaging cell line, but not as significant as in producer cells. We would like to fine-tune the additive concentration and timing in all three platforms to maximize the benefit of using it. However, as you can imagine, the work would include testing a large number of conditions and generating a lot of samples that would need to be analyzed to inform the direction of further round of optimization. Without having a fast in-process analytical methods suitable for non-purified lentiviral vectors, because these are upstream productions, the further process optimization around using the additive X was slowed down. So that's why I'm not able to show you any further results at this moment because of the slow progress of this program. So ideally, we would like to have a fast method that can reliably analyze the crude upstream samples and provide rapid insights to enable us to do timely adjustments to the upstream process of lentiviral vectors. And this is not only applicable to this specific development project we're doing, it is like widely relevant to like all the upstream process development for lentiviral vectors. And in the next slide, I'm showing another example, and this time it's about the lenti downstream process optimization, aiming to further improve the lenti vector recovery and purity. It is very well known in the industry that there is a significant material loss during lentiviral downstream processes. And overall, the recovery is usually just 20 to 30%. Nowadays, we know that in vivo CAR-T and other lenti-based in vivo therapies are being actively developed, and these products have higher purity and quality standards to meet, which means additional purification steps might be needed, and that also means more material loss could happen in the downstream processes. This would actually add to the high vector cost of the cell and gene therapy products, especially that for the in vivo therapies, because they often require higher vector doses. We came across a novel AEX column which claimed high process recovery and residual host cell DNA removal for lentiviral products. So we tested the column using the lenti vectors produced from Soflex packaging and producer cell lines. By just following the manufacturer's instructions without any specific modifications and optimizations, we observed that the vectors from producer cell line had higher process recovery than vectors produced from packaging cell line. We can see that the vectors from producer cell line had a higher titer post AEX, and it also has a lower residual host cell DNA level, but I didn't include the data in this slide. But again, we would like to adjust some parameters to try to improve the process to see the true potential and maximal benefit of using this novel AEX column. But again, analytics became a challenge for us. We didn't have a fast and cost-effective analytical method for lenti vector and purity, and we were not able to get timely feedback from the process to inform further optimization direction. So if we want to do more exploration and optimization, the process would become very time-consuming and labor-intensive. And when there are time and or resource constraints, which is very common in the industry, the optimization work may have to be deprioritized, which was the case for us in this project for testing this novel column, and I'm pretty sure that it is the case for a lot of you in the audience when you wanted to test and try a lot of different new things, but you couldn't because of the resource requirements. So from the above examples, we can see that the producer cell line does offer a great potential for process optimization. And also there are indeed promising process innovations existing in the field available for testing. However, the current analytical toolbox we have for lentiviral vectors may actually become a hurdle for efficiently exploring these potential and innovations. So the analytical methods we usually use, the infectious titer assay, which is the gold standard for lentiviral vectors, but it has a fairly long, like 1 to 2 weeks turnaround time, and also quite high variation across assays because it is a cell-based assay. Alternatively, we can use the particle titer assay via P24 ELISA. That is quicker and a bit more robust, but it still takes a few hours to maybe up to a day to get the results. And it's not really differentiating different particle populations. In our lab, we also try using the dynamic light scattering method to help visualize different particle populations in the purified sample. It works nicely for the purified lentiviral vectors, but it's not working that well for non-purified samples. So it's not really helpful if you want to optimize an upstream process. So to solve all of these challenges, our collaborator Refine team has developed a analytical method based on macro mass photometry, and it can analyze the sample in about 7 minutes. And it is a cell-free, label-free method and direct measurement of the particles, so it's quite robust. It can be used for both the non-purified and purified vectors, and it can measure the different particle populations and analyze the different features of the different particles. So in our mind, as a platform and a process developer, we think this is a very promising method for in-process lentiviral vector analytics, especially for innovative, novel platforms and processes. So Minaris has entered the collaboration with Refine to explore the method, aiming to enable a deeper process understanding and a faster process optimization to enhance lentiviral vector yield and ensure the product quality. So now I'm going to hand over to my co-presenter, Hena, from Refine, to introduce more on the method and how it can help on the LV analytical challenges. Over to you, Hena. Thank you.
Thank you so much, Qian. I'll talk about how is it possible to advance lentiviral vector in process optimization with the macro mass photometry. So let's start by looking at the key challenges across both upstream and downstream processes. So on the upstream side, there's several critical factors directly impact productivity. So we have the transfection efficiency, plasmid ratios, and then we have a cytotoxicity of the plasmids, and they all influence yield and in general cell health. So overall, upstream optimization is about finding the right balance to achieve consistent high productivity. Now, if we shift to the downstream side, the challenges obviously continue. One of the biggest issues is particle loss during purification. And at the same time, lentiviral vectors do not remain stable for a long time during processing. So that creates a constant challenge for purity and yield. We do need to remove impurities, but we need to keep in mind that we are introducing a shear stress through equipment and consumables like membranes, filters, and chromatography columns, so that can further damage the product. So, and then finally, there is always a question of scalability. So if process works at small scale, can we confirm that it's going to perform the same way at the production scale? So now that we outline some of the challenges, again, there are just some of the challenges, the next question is, how do we optimize such a process in a very complex environment? The reality, these challenges are very multifactorial, and addressing them requires analytical methods that are reliable and scalable and robust enough. So many methods currently just fall short. For example, a lot of techniques do suffer from a low throughput. They're impractical for the optimization. Others show high inter-assay variability, which limits the confidence in data and makes that comparison difficult. And this also creates a major barrier for tech transfer, where consistency and reproducibility are crucial. So this raises a very important question: are these limitations coming from the analytical method themselves, or they're inherent to the lentiviral vectors we're working with? So on the top of that, we also have a general issue in the industry, and that is the standardization. While there are ongoing efforts for that, we are still very far from having a universally accepted approaches. So the results in routine monitoring and process control become extremely difficult to implement, and that ultimately slows down the development and the scale up. So we really need a solution that will bring that consistency and robustness and scalability at the same time. So how we can improve it? We need a method that is going to be high throughput, very robust and fast, but ultimately speed and reliability will decrease that cost over time and reduce the optimization time. So the goal is to move from the process development to the production of first batch very quickly. So this is where macro mass photometry can come in as useful. So it does allow results to be analyzed very quickly within minutes. So for example, if you take your sample from your shake flask or chromatography eluate, you are able to get results in a minute. It does allow multiparametric output to be provided very quickly. That means that someone who is analyzing can see the particle size in nanometers, contrast, which is a proxy for mass, and the physical titer. So it's 3 in 1. And the assay is very fast and provides a reliable data output. So now with all of that in mind, you may be questioning what macro mass photometry is. So let me walk you through how the macro mass photometry works. So if you're familiar with mass photometry already, you know that once you put your sample on the glass slide and put it on the instrument, laser shines the light, and that light scatters, and it produces something known as an optical contrast. So you'll see this little dark spots. The darker the spots, the greater the contrast. But macro mass photometry introduces also a Z-sweep. It adds as an output size, as an additional output. So when allowing the Z-sweep, we can see the output is multiparametric. So on the x-axis here, you can see that is the contrast, which is the proxy for mass, because the contrast directly scales with mass, and then size on the y-axis. So how that works and why is it useful for in-process monitoring of lentiviral vector samples? So just to give you a background before going into lentiviral vector samples, that they are very complex and they're very highly heterogeneous, let's start with something a little bit more simple. So when there is a small difference between the particles, whether that's a small difference in size or that's a small difference in the contrast, many methods can essentially find it challenging because they usually rely on just a single parameter. So this is where the multiparametric output becomes critical. So let's see first the graph A. We have two types of beads. They are mixed in the equimolar ratio. And we can see they're the same size, but that contrast, or which is a proxy for mass, it's quite different. So if we are reading this, if we are analyzing the sample and the NTA, we'll see just a size distribution of 100 nanometer, as you can see on the y-axis, the overlapping orange graph. By having this method, we do not only provide which populations are present, but we are able to, for each one of these populations that are the same size but a different mass, able to provide a physical titer. Now let's move on to a slightly different example, which is that graph on the right-hand side of graph B, where we have, again, equimolar mix of two different beads, and they have different size, but they do have an overlapping contrast. Now, as you can see, their size distribution is very close. So methods that do not have a resolving power to distinguish these two populations won't be able to tell you that you have two different populations present in there. So by having the multiparametric output, you're able to uncover differences that would otherwise remain hidden, and that is going to give us a much more accurate and detailed understanding of the sample composition itself. So moving on to lentiviral vectors, now this is where it started becoming a little bit tricky, and that's why it's important to have a very robust analytical control. So it is important to have it to be able to distinguish a true biological variation from the measurement noise, if we can call it noise. So macromass photometry is the method that is going to detect all the particles at the single particle level. So that does not only involve the lentiviral vectors. So some of them, non-viral particles and process-related impurities, are going to be present. And how we know that is by testing the negative control. And in this case, the negative control is a mock transfected cell supernatant. So everything is present besides the viral components. So if you look at the scatter plot on the left-hand side, the blue scatter plot, that is our negative control. So because of the single particle level ability of the instrument, you can see that a lot of impurities are already present. They're not maybe sometimes missed by other analytical methods. And if you look at the right in the middle, the orange scatter plot has that lentiviral vector population present. If we overlap it and look at the contour plot, it's very easy to see that these impurities are still present, but the background impurities are present consistently across the sample. But in the case of your lentiviral vector sample, you're able to see the population in the enrichment. So by incorporating that well-defined negative control, we can apply precise gating, improve the data confidence, and we ensure that we are making decisions based on a real signal, not just doing it arbitrarily. So if we now look and step further and see how this method can allow the real-time monitoring of lentiviral vector productivity and purity across the process, we can start have a look at all of these scatter plots, starting with the crude sample, going into clarification sample, chromatography, concentration and buffer exchange, and then sterile filtration. So on the plot, you can see the gate B on the left-hand side, which is represents our impurities. And in the middle, there is a gate A, which represents our lentiviral particles. Now, again, if we step back and focus what the upstream goal of the process optimization is to increase the viral titer and improve the transfection efficiency. So in that case, we are able to monitor that very quickly and across the board with the macromass photometry. So the advantage here is that all the samples can be taken almost in the real time and tested immediately. As we move to downstream, you can see that the level of impurities are decreasing, and the number that you can see just under the gate A, which is the physical titer, we are enriching the right population. So the goal is of the downstream is to maximize purity while preserving the viral integrity. And here we can see both. We can see how we are getting rid of impurities and that population slightly decreasing in the gate B, and in the gate A, we are enriching. So at the same time, the multiparametric output in this case allows you to track the changes in size distribution, the enrichment of the desired population, and the particle titer. So more importantly, all of this data is delivered on a timescale that is operationally relevant. So instead of running that single experiment a day, you can potentially perform multiple optimization cycles in the same time frame because you're getting results so quickly. And that is the real impact. Faster data will enable you to make the faster decisions or adapt some of your critical process parameters. So it's important to highlight the macromass photometry does provide a physical titer as an output, but how does that work when it's paired with a functional assay? So the key advantage is speed. With macromass photometry, you're getting results in 90 minutes, and as Qian mentioned, sometimes you can wait for the functional data for 1 to 2 weeks. So if we take a look at this example, this is the same data set, just now in numbers, as it is the scatter plot I showed you in the previous example. We can see the consistent trend between the physical titer and the infectious titer. And this alignment is critical because it's telling the physical titer is a reliable early indicator of functionality. So we can also go a step further by looking at the ratio between the physical and infectious titer. In this case, that ratio is below 1,000, which is generally considered very, very good benchmark for the process quality all across the industry. And why does this matter? When you combine physical with a functional titer, you get much better and holistic view of your batch quality. For example, by tracking the ratio, you're seeing where you're actually enriching for functional particles, not just increasing the total particle count. And that you can see the effects of the critical process parameters in general on the productivity. So, for example, in this data set, if you look at the last blue dot, which is our physical to infectious ratio, you can see that there is a slight increase, which means that there is a little bit of a loss of the functionality, although the trend is same, which makes sense because when the lentiviral vectors are passing through a very fine filter, that will introduce a loss in the functionality. So this is while you're waiting for your functional assay results, you can already have a strong indication whether the process is delivering the expected improvements as you are improving or fine-tuning your steps. So that is going to potentially increase the speed of decision-making as well. If we go to a higher level of granularity or just the optimization of the upstream condition, let's see how we can get a quantitative insight across different cell lines. So starting on the left-hand side, we have a scatter plot of 3 different cell lines that Qian already mentioned in her presentation, which are the transient, packaging, and producer cell line. And this is all of these are Minaris samples that we tested. So we can see that going from the transient to packaging to producer cell line, there is an increase in lentiviral population as we move further. But if we want to see very quantitatively, we can start looking at actual numbers. So on the right-hand side, you can see the comparison, the physical titer to the functional titer of these 3 cell lines. So when we compare these 3 cell lines, we can see a very, very clear linear correlation in the increase of the high physical titers in the producer cell line, and they are directly associated with increased effectivity. So even early at this stage, just by comparing these 3 platforms, we can already identify which of these systems produces more particles. And this is really powerful because it allows you to make data-driven decision very early on the platform. And this is also confirming the hypothesis and all the data that Minaris produced over time. And it will allow anyone potentially developing a cell line to test it very quickly. If we go into a practical example of the downstream optimization, now here we just have an example of a downstream optimization on the downstream steps of the producer cell lines, which is again mentioned as a robust and less variable cell line compared to the transgender packaging. So we can see that in the producer cell line, two different samples from the two different fractions of the downstream purification. So the first one is clarified, and the second one is a clarified cell sample after clarification, and the second one is after the AEX chromatography. So we can clearly see here enrichment, even just by qualitatively looking at it. But if we put into the numbers, we almost see a very, very high increase of the particle titer, going from 5E8 to 1.5 to 1.5E10, which is almost giving you like almost a twofold orders of magnitude increase. And this is what we want to see when we are optimizing the downstream process. We want to see that we are increasing the titer, we are reducing the impurities, and we are preserving the viral integrity. And we can see that decrease in the impurities on the single particle level. And so just by evaluating, for example, these two steps, we confirm that there is an improvement after the chromatography, and that improvement, obviously, the concentration is again going to be increased during the concentration and buffer exchange step. And again, we want to avoid the loss of the quality of these particles. So just by using a macromass photometry, even early in the optimization stage or later when we are continuously like tracking the process, we can ensure that every step is going in the right direction. So I'm quickly going to conclude why is the process monitoring important and why this collaboration was so valuable for us and for Minaris. So at its core, macromass photometry provides a unique multiparametric view of lentiviral vectors that are inherently heterogeneous, and we are able to have an insight to their quality. It allows us also to assess the productivity and purity early in development and each stage of the process. It also allows you to monitor across all the fractions going from upstream to the downstream. And as we've shown, you can a sample from virtually any step within minutes and generate meaningful data. So for the lentiviral optimization, where the speed is critical, we can immediately understand the impact of the critical process parameters on both performance and the output of the certain step in the process. So compared to the traditional methods, this will enable us to have a shorter development cycles and the faster optimization, and then earlier insight into process performance. And then if we pair that with the infectivity data, we'll gain a very holistic, high resolution understanding on both yield and quality. And that will allow us to make informed decision in almost real time. So ultimately, this approach enables efficient data-driven path forward to something as complex as a lentiviral development. I'll just walk you through the macromass photometry solution that we used, and the instrument that we used to apply and analyze all the sample from Minaris, that also allow us to validate their data around producer cell line and their performance as well. So we used a Caritra MP, that is a macromass photometer instrument, and is designed to enable analysis on the single particle level. So this has a direct insight into viral purity and ability to monitor stability and integrity. The volume that is needed for testing is only 5 microliters, and it enables running automatically 14 samples in less than an hour and a half. As I was a user of this instrument, I can assure you that it's very, very intuitive and easy to use, and anyone who works as a scientist in the biopharma, we will be able to train him in a lesson a day. And it's, again, complete solution because it's a benchtop instrument with software modules that are very easy, intuitive to use, and the calibrants that are available. Overall, I want to say that this collaboration provided us with a very valuable insight into lentiviral vector development, and it demonstrated how we can contribute to this field and how we can contribute to in-process monitoring of the lentiviral vectors by just improving the speed and the confidence in decision making, and the end of it also decreasing the price by just speeding down the process. So I'd really like to thank Minaris for that collaboration, providing the great samples and the expertise that made all of this work possible. So if you have any questions or you'd like to explore how you can apply this to your own process, Qian and I will be happy to discuss all of it. Thank you so much.